Hello again. Okay, so this is the second video that I'm going to do on variation and diversity. Okay, in the first video we looked at taxonomy, classification, and the binomial naming system, and taxonomic hierarchies, and then a little bit on courtship behaviour at the end. This video is going to be much shorter, just going to do investigating diversity. And how that can be done, and a little bit on types of variation afterwards, and then a couple of examples, and a little bit on standard deviation at the end. Okay, so investigating diversity and differences. This isn't the complete list of ways in which diversity are investigated, but are the ones that you need to be familiar with for the AQA A-levels. So, one method might be to look for measurable or observable characteristics and the frequency in which they are observed. Okay, so this might include looking for physical characteristics, morphological characteristics of organisms to identify them. Now we're going to come back to this in a bit more detail in a later video. So I'm not going to spend too much time on this idea of measurable or observable characteristics, but I will spend a bit more time on these next three. So these are what we refer to as the genetic comparisons. So to make a genetic comparison, we can either compare the sequence of amino acids in a common protein. So there are some proteins which exist, which are that are almost universal, which exist across all species. An example of those might be something like cytochrome C, which is a protein involved in the electron transport chain of respiration. Now that is present in many different species, from bacteria all the way up to multicellular animals, mammals, fish, birds, you name it, they've probably got cytochrome C. So by comparing the amino acid sequence in a common protein, we are able to see how closely related two individuals are. Because we know if they have a very similar amino acid sequence, that will show greater relatedness. Or, going back to that comment that we had earlier on, a more recent common ancestor. Okay, so we could compare the amino acid sequence. We could also compare, I'm going to skip the middle one for a second, but we could compare the base sequence of mRNA. Because I know the base sequence of mRNA determines the sequence of amino acids, because obviously mRNA contains those codons, with each codon coding for a different amino acid. Now this has a higher level of precision, okay, because we know that the DNA code is degenerate. If we know the sequence of codons, we can distinguish between some of those amino acids that might be present in cytochrome C that would have that could be coded for by more than one codon. And then if we want to improve that precision even more, we could look at the base sequence of the DNA. Now, the mRNA we know does not contain the introns, only exons. The DNA is going to be the introns plus the exons. So that has an even greater precision. So the most precise genetic comparison that you can make is by comparing the sequence of DNA. Now, all of these methods rely on the principle that mutations occur at a roughly constant rate. So a mutation would change the base sequence in the DNA. Now that would also change the base sequence in the mRNA and could perhaps change the sequence of amino acids. Okay, so we also know that those mutations that change the base sequence of the DNA are spontaneous and random. Okay, they are always happening. Their rate may increase, but we know over a really long period of time that that rate is pretty constant. So where we see large differences in the sequence of DNA, that means there must have been many mutations. If there are many mutations, we can assume that's going to take a longer period of time. So when we're thinking about how recent or how distant a common ancestor is, if there are very few differences in the DNA base sequence caused by fewer mutations, that would mean there would be fewer differences in the mRNA and fewer differences in the amino acid sequence. And we could assume, therefore, that there is a more recent common ancestor. If there are many differences in the base sequence of DNA, that means there would also be many differences in the base sequence of mRNA. That would perhaps lead to many differences in the amino acid sequence. And we can assume there have been lots of mutations to cause all of those differences. Therefore, we could assume that there was a more distant common ancestor. Okay, so essentially all three of these methods rely on the idea that mutations happen at a fairly constant rate. If there are lots of mutations, there are going to be lots of differences, and we can measure that by looking at the amino acids, or the sequence of DNA, or the sequence of mRNA. Okay, let's move on. So, what causes variation? Well, there may be genetic variation or environmental variation. We've talked about these already. So, mutations that occur will obviously change a sequence of amino acids. We know also from our previous studies that meiosis mixes the maternal and paternal chromosomes in independent segregation, and it mixes the maternal and paternal alleles during crossing over. Random fusion of gametes or fertilization also increase the genetic variation, or is a genetic method for increasing variation and diversity. So all of these, so meiosis and random fusion of gametes, both occur during sexual reproduction. Mutations are happening all of the time. They are spontaneous. And those mutations, really, they're the only things that account for the variety in asexually reproducing organisms. So, asexually reproducing organisms, they don't undergo meiosis, there's no independent segregation or crossing over, no fusion of gametes. So, mutations are what increase the genetic variation. There is also, for example, in bacteria, what we call horizontal gene transfer. Now, horizontal gene transfer only happens in prokaryotes, where they receive genes from the environment or from other prokaryotes directly. Okay, and there's 3 methods that they can do that. So transformation, transduction and conjugation. Now you don't actually need to know any of these. This is all sort of additional information for just for in this PowerPoint. If you want to look them up, you can do. If you want to pause it here and read this, you can do, but it's not going to be asked in your exam, so don't panic about it too much. Okay, so they are the genetic causes of variation. There are also environmental causes of variation. And there is a combination, and these things, the genetic and the environmental causes of variation can be measured. There can be a combination. It's worth noting that a phenotype, which is the physical expression or the observable or measurable characteristic, is produced as a combination of the genetics and the environment. So the phenotype is determined by the genotype and the environment in most cases. However, there are some characteristics, only a few, that are controlled only by genes. Okay, there's no intermediate. An example of that might be the ABO blood groups. So you can either be group A, or group B, or AB, or O. There is no intermediate between these groups. And that's because they are determined by a very limited number, very few genes. Okay, so because there are no intermediate, the data is discrete, and they would be represented on a graph, like so, as a bar graph, bar chart, or a histogram. Okay, there is no intermediates. So if you see this type of graph as a way of measuring the variation, you know that it's controlled only by genes, a limited number of genes, no environmental influence, and that each one of these are discrete with no intermediates. Another example might be earwax, which can be either wet or dry. It can't be damp, if that makes sense. Most characteristics, most phenotypes are controlled by many genes. We call them many genes, poly, meaning many, genic, genes, relating to genes. So most things are polygenic. Okay, they are controlled by many genes and are also influenced by the environment. Now, in this case, we see a continuous variation where there are extremes and means and another extreme, but with intermediates between all of those. Okay, so we can measure those intermediates, put them on a graph, and in a population we get those range of intermediates between the extremes, which forms a normal distribution. Now, if you've watched previous videos, I've talked about normal distribution before, sometimes referred to as the bell-shaped, bell-shaped curves. And we can see it here with the extremes, very few at the extremes, more in the means. This is a representation of continuous variation. So here we've got some other examples. Okay, tongue rolling is determined by just one gene, 2 alleles. You can either be a tongue roller or a non-tongue roller, but you can't be an intermediate. So this is an example of genetic variation caused by just a single gene, so they form those discrete groups. Mass, well, mass is polygenic. There are many genes which determine human mass, and there's obviously the influence of the environment, whether that person has a large diet, whether they do lots of exercise, whether they are influenced by lots of things that could determine or could change their body mass. Okay, so here we see something that's more like a normal distribution. This is an interesting example. So here we have tall and dwarf plants. So these plants, pea plants, these are plants that, that Mendel investigated in his experiments, and he discovered that plants could either be or have the alleles which make them a dwarf plant. They would need to be homozygous recessive for this, so let's say they are that. Okay, don't worry about it if you're not too sure what homozygous recessive means for now. Or they could be tall, and the tall is determined by the presence of at least one dominant allele, like so. Okay, but you may notice that actually both of these groups show a bit of a distribution as well, a normal distribution. And the reason for that is because there are extremes of being a dwarf and there are extremes of being tall. Perhaps these ones down here at the lowest extreme, the availability of light, or water or carbon dioxide was low, and that limited their growth even within that range of dwarf plants. Likewise for the tall plants. These ones here, if they have a limit to the availability of the carbon dioxide or sunlight, water availability, that would limit their growth. Compared to these ones, maybe these have got more high light intensities, more carbon dioxide, able to do more photosynthesis and grow more. Okay, so this is quite an interesting topic. You would see something similar for if you were to measure height in males and females, where we would have height on the bottom, frequency here. Well, females tend to be shorter than males. Okay, there may be some overlap, perhaps, but there would be two sort of distinct groups determined by the genetics of either being female or male, with some influence of many genes, but also influence of their environments. Right, a little bit extra here. So there are some examples, questions that have come up in the past. Not always as straightforward as those examples that I've given before. So we've got a Himalayan rabbit. Its characteristic is black fur. You might think, oh, but it's actually got really white fur. Well, it does, but in fact, that is only because of the inhibition of an enzyme. Now, this enzyme is inhibited at higher temperatures. So where there are higher temperatures, the melanin, which is what produces the darker colour in the fur, the nose and the ears and the feet, at higher temperatures that melanin is not deposited into the fur. Okay, so the fur appears white. It will only produce those black bits in colder temperatures, hence why the sort of the extremes, feet, ears, nose, become black because they are cooler, so the melanin is deposited because that enzyme is less, there's less inhibition of that enzyme. How could you test that? Well, I guess you could shave the rabbit, put something cold on the area, watch the fur grow back, and it should grow back as black. Sounds a bit cruel, but you could do that. Another example, You may be familiar with this, you might not be, but it's very interesting. Many reptiles show an environmental determination of sex. So here we've got an alligator, and it's the sex of those alligators, or the gender of those alligators, is determined by the temperature of incubation in the nest. So in a nest where the eggs are incubated at 33 degrees Celsius or more, so more than or equal to 33 degrees, 100% would be male. At less than 30 degrees or equal to 30 degrees, 100% female. And then in between you get a bit of a mixture. Okay, so it's really important that you RTFQ read the full question. Okay, so just be careful when you're looking at the phenotypes and you're looking at the things that determine those phenotypes, that you consider both the genetics and the environment. Here's the evidence for this bit. Himalayan mouse? I don't know. Okay, so there is an enzyme called, where is it? Tyrosinase. If you have a look down here, it's proposed that normal body temperature of 37 degrees, tyrosinase from Himalayan skin is bound to an inhibitor. Okay, so if it's bound to the inhibitor, that prevents the melanin from being introduced into the fur. At lower temperatures, the affinity of that enzyme to the inhibitor decreases, thus allowing the synthesis of melanin to increase. So hence why we see at lower temperatures in the ears and the nose and the feet that they have black fur. Alligators, environmental regulation of sex determination. We can see that at 33 degrees Celsius, 100% males, 30 degrees Celsius, 100% females. Okay, and actually there has been studies into this, and you'll see that if you read those studies that the depth of the nest is changed to regulate the population size of, or the proportion of males and females in the population. Very interesting. Okay, so we've spoken about this before. Ooh, I don't know why that's there. A couple of other bits to look at for variation. So we can look at variation between different species. Different species. We use the term inter-specific. So ER from different, ER in inter-specific. Within the same species, the A from same, intra-specific. Okay, variation. We also use these terms when we're looking at competition as well. So competition between the same, between different species is inter-specific. Between the same species is intra. We'll come back to that at another time. Okay, so the frequency of measurable or observable characteristics must be must be representative. Okay, it must be representative of the population. So if you want to measure those or observe those characteristics, you need to take lots of samples, so a really large sample size. And I'm sure you've spoken about this with your teachers throughout the year, that it's necessary to have a large sample size to make it representative. There are lots of questions about why many repeats were taken, and we use the word representative. Okay, they must be unbiased. Okay, so it's important that the sampling is random. Oh, it says it there. Okay, so random sampling is really important to prevent bias, to prevent the scientists from introducing any additional variables that could alter the results. Okay, so when we're doing this, we do representative by using a large sample size, and not biased by using random sampling. So, you may have done this at secondary school, you may be familiar with this. These sampling methods, so using a quadrat, are in fact really biased. Okay, you cannot just stand, even I've heard people say before, they throw the quadrat over their shoulder so they don't even know where they're looking. Well, the fact that you're stood in one place in particular immediately adds bias, okay? Because no matter how good you are, perhaps you can't hit every single spot in the field that you're sampling. Okay, so the position where you start would actually add bias. Perhaps if your quadrat lands on these nice daisies here, okay, I'll go and count those daisies. Happy, happy. But if it lands in the mud, perhaps it would prevent you from going to sample there. So we cannot use this method of just chucking a quadrat. So instead, with random sampling, we use a grid system. So a grid is drawn on the field. I'll just show you this here, look. Okay, so this is a sampling collection on a particular area. Okay, a grid drawn, in this case 100 meters by 100 meters. And then random numbers using a random number generator, so you've got them on your calculators, or there are random number generator tables, or there are online tools to create these random numbers. Well, those random numbers can be used to produce coordinates. Okay, so for example here, 5, 15 places it in that position. Okay, or using a random coordinate, 30 by 20 puts it in that position. Now you choose a large number of those random coordinates, then go out into the field, place the quadrat in those positions, record what was in there, and if you have those random coordinates and a large sample size, then we've got that prevents, so the random numbers prevents bias, large sample size makes it representative, and it reduces the probability of chance affecting the findings. And that's a much better way of gathering data. A couple of last bits. We've mentioned a normal distribution in the bell-shaped curve with the phenotype on the x-axis, frequency on the y-axis. And I've also, in one of my previous videos, looked at standard deviation. So standard deviation is the spread either side of the mean. A small standard deviation means there is lots of the population are very close to the mean. A large standard deviation means there's a much bigger spread. One other thing I wanted to point out there, so this is an example of a phenotype, the phenotype being the blood pressure, that measurable characteristic. The mean blood pressure for the diastolic blood pressure is about 82. Okay, with a standard deviation plus one standard deviation plus or minus 10. So most readings fall within the range 72 to 92 milligrams of mercury. Okay, this pressure for the diastolic blood pressure. So we can see that here. Now, one thing I want to point out, this is just a single one standard deviation. Standard deviation works by one standard deviation will include 68% of the population. Two standard deviations we can see here is 95% of the population. Three standard deviation is nearly everybody, 99.7% of the population. I think sometimes referred to as a 68, 95, 99.7 rule. Okay, so I think that is everything that I wanted to cover today. Standard deviation stuff. There is more on this in your mathematical skills booklets that you should have received. Next time, I'll spend a bit more time on genetic comparisons and biodiversity. Okay, thank you.